Welcome back. Please join me in welcoming our next speaker, Dr. Akiko Iwasaki. Dr. Iwasaki is a starting professor of immunobiology and molecular, cellular, and developmental biology at Yale University and an investigator at the Howard Hughes Medical Institute. She's renowned for her pioneering research in immunology, particularly in understanding the mechanisms of innate and adaptive immunity, and her work has significantly advanced our knowledge in combating viral infections and autoimmune disease. And she will talk about the immunology specifically to long COVID. The floor is yours, Professor Iwasaki.
Thank you very much, Stefan. It is my honor to be invited to speak today for Unite to Fight. I'd like to speak about our recent research on immunology of post-acute infection syndromes. Before I begin to speak about these syndromes, I just want to highlight the fact that there are many medically unexplained post-acute infection syndromes that are known to occur after encounter with distinct pathogens. So SARS-CoV-2 joins this list as the newest member. However, there are many other viruses, Ebola virus, dengue, polio, SARS, chikungunya, EBV, and many others that have been reported to cause similar types of outcomes. And non-viral pathogens, like bacterial and parasitic pathogens, have also been reported to lead to these PAIS consequences. And I had this pleasure of writing this review with Jan Chotka, who himself is a ME/CFS patient. And throughout the pandemic, I've been really appreciating how much the patients know about their own diseases and that they're the expert of diseases such as ME/CFS and long COVID. So without their equal participation in our research, we wouldn't be able to make as much progress as we are doing today. So there are many of these infectious agents that can lead to these post-acute phase of infectious disease. But there are common symptoms that are shared by many. These are the core symptoms that include severe fatigue, exertional intolerance, neurocognitive impairment, sensory impairment, flu-like syndromes, dysautonomia, and pain, and many others. So how can these sort of chronic state occur after a so-called acute infection? And there are many theories that have already been discussed on this platform, but I'd like to just sort of talk about 4 simple possible root causes that could be considered and that we are investigating. One is the viral reservoir or persistent virus that you've heard from Dr. Prohl about. And this is, there's so much evidence that's already covered in her great talk, so I'm not going to repeat that. There's also a possibility of autoimmunity, which is well known to occur after following infections such as EBV, leading to multiple sclerosis years down the road. And these kinds of issues may be happening also in people with long COVID. There's also tissue damage and dysfunction that can happen as a result of the acute infection. And this is, what was covered by Dr. Ailey's talk, that the inflammation that persists may be at the root cause of long COVID and PAIS. Another area of sort of emerging consensus is the latent virus reactivation, which is that humans, adult humans like ourselves, we live with multiple latent viruses within us, which are not really causing any diseases at steady state. However, when they reactivate due to some other insults, such as SARS-CoV-2 infection, they could then replicate and cause symptoms. So these 4 root causes can then lead to things like microclot formation, dysbiosis may be contributing, muscle damage, mitochondrial dysfunction, complement activation, and many other downstream consequences that you will and have heard throughout this symposium. So how do we deal with these possible root causes and how do we investigate this? One first of the study that I'd like to share is the collaboration between Dr. David Petrino at Mount Sinai School of Medicine, who is just an amazing investigator. You will hear from him tomorrow. He and I were able to put forces together in the early pandemic to start to investigate the immunology of long COVID. And his brilliant team members, Jamie, Laura, and Dana, contributed to the study. And from the Yale side, we have 6 trainees who also contributed equally: John, Rahul, Pei Wen, Jill, Jeff, and Sasha. And Aaron Ring provided his platform of rapid extracellular antigen profiling, and David Van Dyke his computational biology application. So putting forces together, we are studied patients who have long COVID. These are the patients that Dr. Petrino sees in his clinic. We have 99 long COVID participants, 39 people with convalescent control. These are the people who recovered from COVID but had acute COVID around the same time as the people with long COVID. The healthy control individuals who never was exposed to the virus. And we also had 2 other cohorts, healthcare worker at Yale who contributed various surveys and specimens, and the external long COVID cohort. This is Yale pulmonary long COVID clinic patients who donated their blood for a validation analysis. And the age group, age, sex, and acute COVID severity were as matched as possible with the COVID participants and the convalescent controls. Age hovering around 30 to 58 years of age. Sex is female predominant, and this is female sex is one of the risk factors for developing long COVID. And if you look at clinics around the world, there are more female patients than male patients. We also focused on acute COVID severity being mild, that are not hospitalized. And this is because 90% of long COVID patients start off with a mild infection, and we wanted to focus on that group. And days from acute COVID was hovering around 400 days plus or minus. So this is, well into the first year of their disease. I'm just going to highlight some of the key features, and we have 20 minutes, so I'm just going to go through some of the key features that I'd like to convey today. One is that long COVID patients have elevated type 2 T-cell immunity. So there are at least 3 kinds of immune types, and type 2 immunity is often considered to be optimal for fighting helminths infections, allergy. These are the kinds of immune responses that you see. And for some reason, with people with long COVID, they have elevated levels of cytokines such as IL-4 and IL-6 that's induced in CD4 T cells and CD8 T cells. And if you look at the participants with long COVID, they're really the only ones that have IL-4, IL-6 double positive T cell populations. A second, along with this viral reservoir hypothesis, we see elevated levels of antibody responses to the spike protein in people with long COVID. They're in this purple dots. That's compared to the convalescent controls who don't have disease, and healthy controls are here. And we've only included people who had 2 doses of vaccines just to normalize their exposure to the antigen. So what you see is this elevated levels of antibody, but not necessarily these antibodies are very functional because their neutralizing antibody titers are not elevated, suggesting that they may have persistent antigen for which they're developing higher levels of these anti-spike antibodies. We also looked at the plasma factors, and what we find is that there is elevation of complement factors, C4b, as well as chemokines and cytokines that are elevated in people with long COVID. However, there is one outlier that's really severely associating with long COVID, which is the cortisol level. So the morning cortisol level was reduced in people with long COVID compared to the other groups. And cortisol is a diurnal hormone that's elevated in the morning, and it tapers down during the day. So we wanted to ensure that the collection time doesn't account for this difference in the cortisol level. And in fact, there is no difference in the collection time of the blood draw. This is a Z-score showing the level of cortisol in circulation. And you can see that long COVID patients had much lower levels than the healthy control, the convalescent control. Because this phenotype was striking, we also recruited this external cohort from Yale Pulmonary Clinic. And those participants also had reduced levels of cortisol in the morning. So cortisol is a very important hormone. It's known as a stress hormone, but it actually has a lot of physiological functions that are important for daily lives. The cortisol level that we see that's reduced could be coming from the lower levels of adrenal glands that are able to provide the cortisol, produce the cortisol. But they do so in response to the ACTH hormone that's secreted by the pituitary, which is actually in turn induced by the CRH hormone that's released from the hypothalamus. So there's a very nice triangle of hypothalamus, pituitary, adrenal axes that control the level of cortisol. Because this level was lower, we wanted to understand if the ACTH level might be elevated to compensate for the low levels of cortisol in patients. But that's not what we found. Long COVID participants had just as normal level than the healthy controls and convalescent control of the cortisol, ACTH level in circulation. So this led us to think that perhaps the defect is not at the level of the adrenal glands, but potentially in the hypothalamus or the pituitary that control the level of cortisol through the adrenal glands. We also looked at latent virus reactivation, and this was enabled by the REAP strategy that Dr. Ring's laboratory developed. So he included 200 plus viral antigens in his REAP library to be able to analyze IgG reactivity against these viruses. And what we found was that there were 3 antibodies that were elevated in people with long COVID, and these antibodies reacted against Epstein-Barr virus glycoproteins, P23 and GP42, as well as varicella zoster virus, GE. And because the seropositivity for these viral pathogens were not different between people with and without long COVID, we suspect that this elevated level of IgG represents recent reactivation with EBV or VZV. And we wanted to ensure that this measurement with REAP can be validated by 2 other orthogonal approaches. So we used Seraimune, which is a phage display library, as well as ELISA to validate these findings. And indeed, it does validate with 2 different methods. So what we are seeing again here is the elevated level of IgG against the Epstein-Barr virus P23, GP42, and VZV GE proteins. And interestingly, the level of the EBV reactive IgG correlated with these cytokine secreting CD4 T cells that I mentioned earlier. So whether it's a cause and effect, whether it's because of these type 2 immunity that these patients are reactivating EBV, or EBV is now inducing this type 2 immunity, we don't know what the relationship of this correlation is yet, but that's something we're investigating now. So, all in all, when we look at the, we asked David Van Dyke, who's a computational biologist, to come up with a handful of immunological factors that can distinguish people with long COVID or without long COVID. And in fact, just a handful of immune factors such as the circulating level of dendritic cell subsets, CD4 memory cells, galectin-1 and ABV reactive antibody, as well as, of course, the cortisol, were able to separate people with long COVID with 94% accuracy. So this led us to the realization and understanding that there's biological mechanisms that can explain long COVID, and that we may be able to interfere with some of these pathways if there is a causality associated with these factors. So we took the same data set and now analyzed these data in terms of female versus males with long COVID. And this study is led by Julio, Takehiro, Jamie, Laying, and David, with many other collaborators that are listed. So this analysis provided some very interesting insights. So looking at the relative frequency of different symptoms that are reported from people with long COVID, there were some symptoms like sleep disturbances or fatigue and nausea that were equally reported from males and females. There were some other female predominant symptoms such as dizziness, body temperature issues, cough, chest pain, and so on. And most sexually diverse reporting was observed for a sexual dysfunction for males and hair loss for females. So just looking at the set of symptoms, you can already tell that the disease may be quite different between males and females. And in this study, there's a preprint already published there, but one thing to highlight is that Julio found that lower levels of testosterone was the top hormone predictor for long COVID status in females. So as you know, testosterone is a very important hormone that regulates many different reproductive function, as well as other physiological functions. And males have elevated levels compared to females. Females with long COVID have reduced levels of testosterone, and that is the per unit changes in the testosterone turned out to be the top predictor for long COVID status. And conversely, in males with long COVID, Julio found that estradiol levels, changes in estradiol levels, was a top hormone predictor for the long COVID status in males. So these studies are now leading us to the understanding that there is a more broader than just cortisol hormonal dysregulation that's happening in people with long COVID. And that sex disaggregated analysis is now revealing unique features associated with disease as well as potential drivers. And autoimmunity, this is something that we haven't even published yet on medRxiv, but we are now investigating the role of autoantibodies. And this is a work that's led by a brilliant postdoc, Kayla Sa, in the lab, where she purified immunoglobulin, IgG, from healthy people, convalescent controls, acute severe COVID, these are hospitalized patients, and long COVID participants. And then she transferred these IgG into animals. These are just normal wild-type C57 Black 6 mice. And then she carries out a battery of behavioral tests. And many of these tests show no differences whatsoever. However, there are 3 tests that really stood out. One is a rotorod test. This tests the balance and coordination by putting the animals onto this rotating rod, and you measure the amount of time it takes for the animals to fall off. So the animals that received IgG from long COVID participants fell off the rod much faster than those that received healthy control or PBS. Second thing that she detected is a difference in the grip strength force. So this is a strategy where you let the animals hang on to the grid and you pull until the animals fall off the grid, testing for muscle strength. And this clearly demonstrates that IgG from people with long COVID, when injected into animals, had a severe impairment in the muscle strength. And finally, she tested the thermal sensation, pain sensation, using the hot plate test. And here again, IgG from patients with long COVID had a lot faster latency to pain behavior, meaning that they had to withdraw their paw much quicker than the control animals. So this overall is leading us to this understanding that autoantibodies, or at least cross-reactive antibodies that are injected into animals alone are sufficient to cause the thermal sensation, pain sensitivity, as well as reduced muscle strength and reduced coordination and balance. And finally, just to quickly touch on the tissue damage and dysfunction, this is a work that we carried out with Professor Professor Michelle Monge's team at Stanford, where we used a mild respiratory-only COVID model. This is an animal model that we developed earlier in the pandemic, where we can control the organ where the virus replicates by introducing human ACE2, the viral entry receptor, only in the lung. So these animals, there is no possibility for them to become infected in the brain. And yet, when we infect those animals with a very mild level of SARS-CoV-2, and then follow them over time within 7 weeks, we see a chronic change in the brain and damage that's associated with this acute infection. So this is a summary that I'm borrowing a beautiful figure from a review that was written of this study, where basically what we demonstrated is that this mild respiratory-only COVID results in changes in the brain. There's a neuroinflammation that's occurring in the subcortical white matter, which is mediated by chemokines and other cytokines, that then results in elevated reactive microglia. And that state of microglia has significant impact on the surrounding cell, such as the oligodendrocyte precursors and the myelination that they provide. So we see loss of axon myelination around this region, as well as in the hippocampus. Similar kinds of reactive microglia elevation is detected, and there also we see hippocampal neurogenesis being reduced. And this is accompanied by studies with autopsies from human brains and other MRI studies that corroborate these findings. So this illustrates that direct infection of the brain is not required to induce significant CNS pathology and neurocognitive dysfunction. So our findings and many others who collaboratively and collectively really demonstrate that there are significant changes occurring in people with long COVID, such as reactivation of EBV, autoantibody production, hormonal dysregulation, and sex differences in these features. So what do we do now that we have some understanding of these changes? Well, I think what we need to do now is move forward to developing therapy by randomized clinical trials, such as the one that Dr. Ely already described. But we can target these root causes with different drugs. For instance, autoimmunity, there are many very successful drugs that are already developed for autoimmune diseases that we can repurpose to treat patients with autoimmunity. We have completed enrollment for the Yale Paxlovid trial. This is, again, a randomized clinical trials using 15-day course of Paxlovid in people with long COVID plus a placebo arm. And now we're just collecting the last set of data to be able to unblind ourselves in midsummer. So we'll find out what's going on there. But the important thing is that we don't expect everyone to be cured by Paxlovid, but rather what we're going for is we want to understand who is benefiting from Paxlovid. Can we find biomarker of benefit from Paxlovid so that we can treat patients with those biomarkers going forward to improve their health? You already heard from Dr. Eli, so I'm not going to talk about this particular trial. We're also doing an antiviral trial against latent virus with David Petrino and Amy Prohl. And this is my last slide. I wanted to just thank the amazing people who I have the pleasure of working with every day. And I feel like we're already united to fight against these diseases, but this Unite to Fight here today is really bringing this at the global scale. So I'm really excited to speak with all of you, and I'm happy to take questions.
Thanks a lot, Professor Iwasaki. And sorry for cutting you short, but we have so many questions waiting for you. And the first one would be, do you think the symptoms are most likely caused by some ongoing process that can be reversed? So viral persistence, misfiring immune response, inflammation, reactivated viruses, etc. Or is it more likely an irreversible damage to the body from the initial infection or something else that happened after that?
I strongly believe that these processes, some of these processes can be reversed. For example, if persistent virus is driving the disease, antivirals or even monoclonal antibody cocktails against the virus should be able to remove the source of the problem. And once that happens, hopefully that tissue will regenerate and restore to normalcy. And similarly for autoimmune diseases, we may be able to get at the source of the problem, which may be the autoreactive B cell secreting antibodies. EBV, again, if we can get rid of the reactivated virus, can we restore health? So all of these things, I don't think it's an irreversible damage. And we're actually doing experiments in mice. We develop mouse model for everything. We are doing experiments in mice where we are seeing some reversal of damage, for example, from the chronic inflammatory conditions that occur in the brain. So I think, I'm not going to say that we can do this for everyone, but I think there is a path forward to reversing this.
Thanks a lot. And we had one question at Wes Ely's session that we brought into this session, and it's about interferon therapy. It seems to be a mixed bag, but what is your opinion as a potential way to treat post-acute infection syndromes, or at least specific cases accompanied with latent viruses?
Yeah, so it is a complex one because we know, for instance, that early interferon is quite beneficial during the acute severe COVID, whereas late interferon is actually pathological. So we need to first identify the right kinds of people. The people who might benefit from interferon therapy are perhaps the immunocompromised people who, with known replicating virus that cannot be treated with other means like antivirals and monoclonals. There, we had actually published a study in rag knockout animals. These are animals that lack the adaptive immune system, which have chronic replication of SARS-CoV-2. We were able to treat this mice with a one shot of RIG-I agonist, which induces interferon and clears the virus. So there is some evidence that chronic replicating virus could be treated with interferon, and that's what happened with other chronic viral infections. So if we can identify those people who might benefit versus people who are having autoimmunity, you definitely don't want to inject interferon into those people. That might exacerbate the disease. So again, knowing who has what is going to be very important.
Thanks a lot. And maybe a last question. What about lab work that you recommend for long COVID and ME/CFS, especially for immunological lab work?
Yeah, so even though our machine learning did identify some of these factors, we really need to do this at a larger scale to be able to say, okay, here's a diagnostic tool that can be developed for everyone with long COVID or ME/CFS. Because the drivers are different, say that, these 4 different drivers that I described, there may be combinations, right? That some may have replicating virus plus autoimmunity. So unless we can identify like a panel of biomarkers that can distinguish people with each of these drivers, it may be difficult to say, okay, here's a diagnostic test. But I think it can be done. It just has to be maybe like an array, a test array, where people can test multiple different drivers. But this is something we're working towards.
Yeah, thanks a lot for your enlightening presentation and for the sneak preview into your unpublished work. It's always inspiring to hear about your science. Thanks a lot. And yeah, bye.